Welcome IB Biology students. This is your 2.3 carbohydrates and lipids video notes. Remember to write down any questions you have as well as take your notes as you go along. So the first thing that we want to remember from both chemistry and our introductory biology class in 9th grade is that organic compounds contain carbon and are found in living organisms. And we have several different types of organic compounds that are found inside of living organisms here: glucose, ribose, different amino acids, and then of course glycerol and fatty acids. But just because it contains carbon does not guarantee that it is going to be an organic molecule. There are a handful of carbon-containing compounds that are inorganic. You have three different categories of inorganic carbon-containing compounds. You have oxides of carbon, which would consist of your carbon monoxide and your carbon dioxide. You also have your hydrogen carbonates. So your hydrogen carbonates, your first one that you have here is hydrogen carbonate ion. This is really important in pH buffering. Your second one that you have here is sodium bicarbonate or sodium hydrogen carbonate. It's called both things, but you commonly refer to it as baking powder. In our carbonate category, so very similar to hydrogen carbonates, but just don't contain that hydrogen, we have the first one, which is just our basic carbonate. This is what is found in rocks. Your next one, which is calcium carbonate. This is what forms shells or exoskeletons. And then you have sodium carbonate, and this is baking soda or baking ash. So baking powder has this hydrogen, and baking soda has no hydrogen. It's just the sodium and the carbonate. All right, so the next thing we need to do is remember what our definitions for mono, di, and polysaccharides are. Remember that a saccharide is a sugar, so we're focusing just on the sugars on the carbohydrate section of these notes. So we have different mono, di, and polysaccharides for both animals and plants, and we need to remember that. Monosaccharides, mono means one, so monosaccharides would be a single sugar. They're very small, they're easily absorbed sugars, so they're very fast release for respiration. I apologize for that. So this is glucose and galactose in animals, and then in plants this is fructose. So fructose is also a monosaccharide for fast release during respiration. I apologize for that. The next category we have are disaccharides. Di means two, so these are sugars made of two sugars. So lactose and maltose. Disaccharides are really important because they're quickly digested into monosaccharides. That bond can be quickly broken. Lactose is what is found in milk, so it is a very ideal sugar for nursing young because it is a disaccharide and that bond can be broken very speedily. You also have sucrose in plants. This is also a soluble but unreactive sugar, so this is important for the plant because it can be transported around the plant in the phloem. The last category we have are polysaccharides or mini saccharides. Polysaccharides, such as in animals we have glycogen, are insoluble storage molecules, mainly due to their size. This is important because our excess sugars can then be converted later by insulin for storage in the liver. In plants, you have both cellulose and starch, and you have two different types. Cellulose, as we remember, is what makes the plant cell wall. But starch is what is the energy storage molecule for plants, just like glycogen is the energy storage molecule for animals. All right, so most monosaccharides or single sugars have a ring structure when they are in a water or aqueous environment. Here are two common ring structured monosaccharides. We have glucose over here and ribose here. If you look, glucose is a hexose sugar and ribose is a pentose sugar. Most students believe that these names, pentose and hexose, has to do with their shape. While it does correlate to their shape, that's not actually how the name is gotten. The name relates to how many carbons make up that structure. And the way that we count the number of carbons is we start here at the oxygen, and then we go around in a clockwise manner and just simply count the carbons that we come in contact with. So here your first carbon is 1, 2, 3, 4, 5, and 6, which means it is a hexose sugar. Ribose, on the other hand, if we start at that 0, you get 1, 2, 3, 4, 5 carbons, which means you have a pentose sugar. So here, looking at some others, we have galactose and fructose. So you can see fructose is another pentose sugar, and galactose is another hexose sugar. Glyceraldehyde is also a monosaccharide. It is a monosaccharide that does not form a ring structure in water. So not all monosaccharides do, but many do. All right, if we remember from our 2.1 molecular biology notes, we've already learned about condensation and hydrolysis, but now we're going to be more specific when we're talking about the carbohydrates, and then later we'll put this in term with the lipids. So just as a review, condensation makes bonds. It releases water when it makes bonds. The condensation reactions are also anabolic reactions, which means we are building molecules. Whereas hydrolysis, we're splitting water, we're breaking bonds, and they are also catabolic reactions, so we're going to break down molecules. So just a quick review of the condensation and hydrolysis reactions. I'm going to take you back to that website link. All right, so let's watch the animation and review condensation and hydrolysis.
A condensation reaction joins two molecules together to form one larger molecule. An enzyme removes a hydroxyl group from one molecule and a hydrogen atom from another, then speeds the formation of a bond between the two molecules at their exposed sites. Typically, the discarded atoms join to form a molecule of water. Hydrolysis is a type of cleavage reaction. It's like condensation in reverse. An enzyme splits the molecule into two parts, then attaches a hydroxyl group and a hydrogen group from water to the exposed sites. The result is two smaller molecules. Condensation and hydrolysis are used to put together and break apart many biological molecules.
All right, so that was a nice little review. Here's another way to write it. We can go from our monomer, or monosaccharides in this case, and through our condensation reaction, make a dimer or a disaccharide. If we do another condensation reaction, we could make a polymer or a polysaccharide. If we go in the reverse, we can go from polysaccharide through hydrolysis to a disaccharide, and from a disaccharide through hydrolysis to a monomer. So this is a really great simplified review of the processes and how they build molecules and break down molecules. So let's talk about specifically how does this happen with those saccharides. So if we take glucose and we add another glucose through a condensation reaction, we'll get maltose, which is a disaccharide, and of course our water molecule. So when we look at this, we're going to take our two glucose molecules. Here is where the bond will be, okay? We're going to go through that condensation reaction. We're going to make the water as the byproduct, and then this bond is called a glycosidic bond. So glycosidic bond is the bond formed between two saccharides within a larger molecule. Monosaccharides are always found with their carbohydrates in a 1-to-1 ratio. If you remember that from the 2.1 notes, ribose, which is C5H10O5, so it is so you can see there's that 1-to-1 ratio. Glucose, you can see here if we were to write glucose out, it is C6H12O6, which is also in the 1:1 ratio. All right, but you don't have to use always glucose plus glucose. What happens if you put glucose and galactose together? If you do glucose and galactose, you get lactose. If you do glucose and fructose, that's how you make sucrose. So you can do a condensation reaction between any of the combinations of the glucose, galactose, and fructose molecules and get a different disaccharide each time. All right, looking at our polysaccharides. Polysaccharides, remember, are just molecules of, or polymers of more than 2 molecules. They can be long or branched. So the bond that we just showed in the other slide was a carbon 1 and a carbon 4 bond. So that glycosidic bond was between the 1 and the 4 carbons. Glycosidic bond can also be between the 1 and the 6 carbon. If you have a 1,4 glycosidic bond, you're going to have a straight chain, such as this part here, which would be all 1,4 bonds, also here. But here in our glycogen, you can see a branched or a bent chain, and that's because this molecule is forming a 1,6 glycosidic bond, and that's going to give us a bent chain. So you can see examples of starches, glycogens, and celluloses. All of these 3 are showing the 1,4 bonds, but you can see over here that glycogen is made up of both 1,4s and some 1,6s. There are 3 main types of these polysaccharides. These are the ones that are listed here: the cellulose, the starch, and the glycogen. Cellulose is always an unbranched polymer. It is the orientation of the glucose units alternate. It's either up or down, so the glucose kind of flips around. But this is going to allow the polymer to be straight and to be arranged in parallel lines, with the H bonds, and that's going to give strength to the cell wall. Think about laying cellulose on top of each other in nice straight lines. That's going to give lots of strength to the cell walls. Starch, on the other hand, can have 2 different forms. It can have a form called amylose or a form called amylopectin. And it's really important that you know which one is which. Amylose is only the 1,4 glycosidic bonds, so it is only going to be straight. It's not going to be branched. Whereas amylopectin is going to have some of the 1,6 linkages, so there will be some branching in that form of starch. Glycogen is very similar to amylopectin, but it has more 1,6 linkages, so there's more branching. Both forms of the starches that we just talked about, the amylose and the amylopectin, are hydrophilic, but they're too large to be soluble in water. Concentrated, it's really important that glucose and other monosaccharides be made into polysaccharides for long-term storage, and that's because concentrated glucose storage in a cell would cause too much water to enter the cell via osmosis. If you have too much water entering the cell via osmosis, then the cell could rupture or burst, and that would lead to apoptosis or cell death. So it's really important that our glycogen and our starch are able to be formed and used as storage. Even though they're hydrophilic, they are too large to be soluble in water, but they don't cause that increase of water into the cells. All right, so just a summary of our condensation to hydrolysis reactions. When we're talking about carbohydrates, we're going to go from a monosaccharide to a disaccharide, making water through condensation, from a disaccharide to a polysaccharide, again making water through condensation. If we were to break down those polysaccharides, we would have to input water for a hydrolysis reaction, then we would get a disaccharide, input more water in another hydrolysis reaction, and we would get the monosaccharide. All right, so that is it for our carbohydrate section. Now let's move on to lipids. Remember that lipids are carbon compounds, and they are mostly or entirely hydrophobic or insoluble in water. You have three main types of lipids. You have triglycerides, which are made of three fatty acids, which is the tri, and the one glycerol. These are also typically your fats and your oils. Your three fatty acids become what we call a hydrocarbon tail, and that is mainly what makes them hydrophobic. Phospholipids, we've talked about at length in introductory biology, are important in cell membranes. They have two fatty acids instead of three, like a triglyceride. They do have one glycerol, but they also have a phosphate group. And because they have that phosphate group, they are partly hydrophobic. Only the fatty acid and glycerol aspect are hydrophobic. The phosphate head makes that part hydrophilic. So they're very important in forming membranes of our cells. We also have steroids. Steroids are lipids that have four fused rings. You can see one here. So we still have that ring structure like we had in carbohydrates. Some examples of steroids are cholesterols, which is why we always kind of drew them in this kind of shape when we were doing cell membranes in introductory biology. They're also progesterone, estrogen, and testosterone are steroid examples. Let's look at a condensation reaction. Here we have three fatty acids, and you can see this is that line structure, and you can see here why it's so helpful to draw that line structure. We want to remember that each of these bends or junctions is where a carbon and two hydrogens are attached, so it makes it a little bit easier to write. And then we have our glycerol molecules, so we're going to make a triglyceride through this condensation reaction. So this is going to be where our bonds are formed. You can see we have enough of these molecules to our elements to form our water. So when we're looking, we're going to make one water for each. You actually, to make one triglyceride molecule, you actually have to do three condensation reactions. The bond that forms from a condensation reaction within a fat or a lipid is called an ester bond. So glycosidic bond is for carbohydrates. Ester bonds are for triglycerides. All right, so energy storage. Let's talk about the difference, what types of things we use for energy. So lipids are better as long-term energy storage, and this is because energy is released from these lipids is twice that of the carbohydrates. It's mainly because there are more bonds to break. All right, stored lipids also have secondary roles. They can be heat insulators or shock absorbers. They're heat insulators because they are poor conductors of heat. Since they are poor conductors of heat, they keep the heat inside the body. This is also why a lot of our fat is stored as adipose tissue just underneath the skin. They're also shock absorbers because lipids or fats are liquid at our body temperature. So that allows them to be a shock absorber. That also is the reason that we have fat tissue or adipose tissue around our kidneys and some of our other organs. All right, let's look at glycogen versus glucose. I apologize, it's not really glucose. It is really fatty acids. So this is glycogen versus fatty acids. All right, so glycogen as an energy storage, remember that glycogen is a carbohydrate. We store glycogen in both the liver and some of our muscles. It can be broken down into glucose rapidly, and the key with glycogen is it can be used in anaerobic and aerobic respiration. Fatty acids, on the other hand, although we get more energy when we do break them down, they can only be used in aerobic respiration. So we can't get to them as fast as we can with the glycogen. All right, so we've also learned in introductory biology and chemistry that you have saturated fats and unsaturated fats. Saturated fats are formed when we have single bonds only. Saturated fats have single bonds and are solid at room temperature. These are typically animal fats. Unsaturated fats have double bonds. They don't have to have a lot of double bonds. They just need to have one. Here is an example of a double bond. See how there is just one double bond in one of the fatty acid chains? By having only one double bond, this makes them an unsaturated fat. These are typically plant oils and they are liquid at room temperature. So now, looking at this example, can you deduce the meanings of both mono unsaturated and poly unsaturated? All right, now that I've given you a little bit of time to think about it, of course, mono means that there is only one double bond, and poly means that there are two or more double bonds. Some interesting information is that omega-3 fatty acids have a double bond in the third from the end terminal. So an omega-3 would have a double bond in the 1, 2, 3 right here. This would be an omega-3. Omega-6 fatty acids have the double bond in the 6. So the 1, 2, 3, 4, 5, 6, that is where our double bond is. So this would be an omega-6. You also have something called a cis unsaturated. That's when the hydrogens are bonded to the carbons on the same side, and then a trans when they're bonded to carbons on the opposite sides. When we get to cell membranes, we're going to do some more work with this cis and the trans, and I'll show you in class as well. Unsaturated fats are artificially produced, can be artificially produced, and they're actually banned in many countries. They're banned in many countries because there is a positive correlation between the trans fats and coronary heart disease. Saturated fatty acids are naturally occurring in animal fats and some vegetable oils. And although there is a positive correlation between the saturated fats and the coronary heart disease, it's not fitting with all the population. So therefore, we can say or confidently say that saturated fats probably don't cause coronary heart disease. But because those trans unsaturated fats, so those hydrogens bond to carbons on the opposite sides, do have a correlation between with coronary heart disease, that is why they are banned in a lot of countries. All right, so when we're looking at this research, such as the research between the trans unsaturated fats and the coronary heart disease, we want to look at the data and ask ourselves some questions, two questions mainly. And the first one is we want to look at implications. Do the results of the research support the health claim strongly, moderately, or not at all? And some of the ways that we can determine this is we can look and see, is there a correlation between the intake of lipids and the rate of the disease or benefit? We want to look at the difference between the average rate of the disease with different levels of lipid intake. We want to look at how widely spread the data is. The data is typically presented on a scatter graph or scatter plot, or we can look at the error bars. If we're looking at a scatter graph, is the data more widely spread on the scatter graph? If we're looking at error bars, are the error bars taller or how tall they are versus much smaller? That's going to tell us how widely spread the data is. We also want to look and see if the statistical tests that were done show significant differences. So these are all questions that we would ask ourselves to analyze the data and be able to determine what the implications of the research are. The second thing we want to look at are limitations. And these is going to be were the methods rigorous, or were there weaknesses in the method that may cause uncertainties? All right, so Miss Julie has lots of typos in this slideshow. I do apologize. All right, so were the methods rigorous, or were there weaknesses in the methodology that could cause uncertainties? Some of the common things that you want to look at is you want to look at the sample size. Was it a large enough survey or sample size to be able to cause any, to be able to draw any conclusions? You also want to look at the distribution of sex, male, female, of the age range of the participants, the state of health initially, the lifestyle choices of those people. If there was an uneven sampling, it's okay, but then you need to look to see if the researchers took any adjustments or if they made any adjustments. And the last thing you want to look at is, were the measurements of the lipid intake reliable? A lot of times this data is presented or collected in a survey format, and how confidently can you ensure that all 100,000 of your participants recorded the amount of lipids that they took on a survey? All right, so that's it for today. There's a lot of new information. I hope it wasn't too overwhelming. Please remember to bring your questions to class so that we can look over them, and I will show you some models of the difference between a cis and a trans unsaturated fat so that you can see what they look like. Have a great day.